Hello friends, warm welcome in this video, that is the innate immunity. Friends, in the earlier video we have learned salient features of innate immunity and the various aspects of anatomical or physical barriers. Friends, no doubt the anatomical barriers provides a strong defense to our body, even though some pathogens have evolved strategies to penetrate these defenses, and the epithelia or the skin may be disrupted by wounds, abrasions, and insect bites that may transmit pathogens. Once the pathogen enters through these epithelial barriers into the tissue spaces of the body, a number of cellular receptors and soluble proteins that recognize these pathogen components play the essential roles of detecting the pathogens and triggering effective defense against these. Thus, the phagocytic cells make up the next line of defense against the pathogens that have penetrated through these epithelial cell barriers. Macrophages, neutrophils, and dendritic cells in the tissues and monocytes in the blood are the main cell types that carry out phagocytosis. So, what is phagocytosis? Phagocytosis is nothing but the cellular uptake or eating of a particulate materials such as bacteria, a key mechanism for eliminating pathogens. So, is this mechanism is present in only human body? No. This mechanism is present even in invertebrates also. The major role of the cells attracted to the site of invading organism is evolutionarily ancient and present in both invertebrates as well as vertebrates. Ilya Mechnikov initially described the process of phagocytosis in 1880 using cells from starfish, which belongs to echinoderm invertebrates similar to vertebrate white blood cells, and ascribed phagocytosis a major role in immunity. Most tissues contain resident populations of macrophages that functions as sentinels for the innate immune system. Through various cell surface receptors, they recognize microbes such as bacteria, extend their plasma membranes to engulf them, and internalize them in phagosomes. Phagosomes are the endosomes which are results from the phagocytosis. Lysosomes then fuse with the phagosomes, delivering agents that kill and degrade the microbes. Neutrophils are the second major type of phagocytes, usually recruited to sites of infection. Finally, the dendritic cells also can bind and phagocytes microbes. Uptake and degradation of microbes by dendritic cells play key roles in initiation of adaptive immune response also. In addition to triggering phagocytosis, various receptors and phagocytes recognize microbes and activate the production of variety of molecules that contribute in other way to eliminating infection. So how does the phagocytic cells recognize microbes and initiates the phagocytosis? Phagocytic cells express variety of receptors on their surface. Some of these receptors directly recognize specific controlled molecular components on the surface of microbes, such as cell wall components of bacteria and fungi. These conserved motifs usually present in many copies on the surface of pathogen, that is bacterium, fungal cell, parasite, or virus particle, and thus are called as pathogen associated molecular patterns, or in short PAMPs. PAMPs. These pathogen associated molecular patterns can be expressed by microbes whether or not the microbes are pathogenic. Thus, many researchers now started to use the more general term microbe associated molecular patterns instead of PAMPs. The receptors that recognize PAMPs are called as pattern recognition receptors, in short PRRs. Some pattern recognition receptors that bind microbes and initiate phagocytosis of the bound microbes are presented here. So here the pattern recognition receptors are divided into two groups: C-type lectin receptor, that is called as CLRS, and scavenger receptors. The CLRS contains mannose receptor, dectin-1, DC-sign. Mannose receptor recognize mannans which are present on bacteria, fungi, and other parasites. Dectin-1 recognize beta-glucans from some bacteria and most of the fungi, whereas DC-sign receptors recognize mannans from bacteria, fungi, and parasites. Scavenger receptors are of two types, SRA and SRB. The SRA receptors recognize lipopolysaccharide, lipoticoic acid from the bacteria, whereas SRB receptors recognize lipoticoic acid, lipopeptides, diacylglycerides from the bacteria and beta-glucans from the fungi. Most of the pathogen-associated molecular patterns that induce phagocytosis are cell wall components, which includes complex carbohydrates such as mannans and beta-glucans, lipopolysaccharides, and other lipid-containing molecules, peptidoglycans, and surface proteins. Activation of phagocytosis can also occur indirectly by phagocyte recognition of soluble proteins that have bound to microbial surface, thus enhancing phagocytosis, a process which is called as opsonization. This word is derived from the Greek word which means to make tasty. Many of these soluble phagocytosis enhancing proteins called as opsonins also bind to conjugate repeating components on the surface of the microbes such as carbohydrate structures, lipopolysaccharides, and viral proteins. Hence, they are sometimes referred to as soluble pattern recognition proteins. Once bound to microbe surface, opsonins are recognized by membrane opsonin receptors on phagocytes, activating phagocytosis. A variety of proteins functions as opsonins. Many play other roles as well as in innate immunity. For example, the two surfactant collectin proteins SPA and SPD are found in the blood as well as in the mucosal secretions in the lungs and elsewhere, where they function as opsonins. After binding to microbes, they are recognized by CD91 opsonin receptors and promote phagocytosis by alveolar and other macrophages populations. These functions of SPA and SPD contributes to clearance of fungal respiratory pathogen Pneumocystis carinii, a major cause of pneumonia in individuals with ears. Mannose binding lectins, that is MBL, a third collectin with opsonizing activity, is found in the blood and respiratory fluids. L-fucolin, a member of the fucolin family that is related to MBL and other collectin, is found in the blood where it binds to acetylated sugars on microbes including some streptococcal bacteria. The complement component C1q also functions as opsonins, binding bacterial cell wall components such as lipopolysaccharides and some viral proteins. MBL, fucolins, and C1q share structural features including similar polymeric structures with collagen-like shafts, but have recognition regions with a different binding specificities. As a result of their structural similarities, all are bound by the CD91 protein receptor and activate pathogen phagocytosis. Another opsonin, which is often called as C-reactive protein CRP, recognizes phosphorylcholine and carbohydrates on bacteria, fungi, and parasites, and is then bound by FC receptors for immunoglobulin G found on most of the phagocytes. FC receptors also are important for the opsonizing activity of IgA antibodies and some IgG antibodies subclass. After binding specifically to antigens on microbe surface, the FC regions of these antibodies can be recognized by specific FC receptors, triggering phagocytosis as an important mechanism by which adaptive immune response clears antigens, opsonizations by antibodies. The most effective opsonin are several components of the complement system which are present in both vertebrates and invertebrates. Complement straddles both the innate and adaptive immune system, indicating that it is ancient and important. In vertebrates, complement consists of more than 30 binding proteins and enzymes that functions in a cascade of sequential activation steps. It can be triggered by several innate soluble pattern recognition proteins as well as by microbe-bound antibodies generated by the adaptive immune response. Phagocytosis is one of many important antimicrobial effects resulting from complement activation. Individuals with MBL deficiencies are predisposed to several respiratory tract infections, especially pneumococcal pneumonia. Interestingly, MBL deficiencies may be protective against tuberculosis, probably reflecting MBS opsonizing role, enhancing the phagocytosis of a Mycobacterium tuberculosis, the route by which it infects macrophage, potentially leading to tuberculosis. The binding of bacteria, fungi, protozoan, parasites, and viruses to phagocytes via pattern recognition receptors or opsonins and opsonin receptors activate signaling pathways. These signaling pathways trigger actin polymerization, resulting in membrane extension around the microparticles and their internalization, forming phagosomes. The phagosomes then fuses with the lysosomes and in neutrophils with preformed primary and secondary granules. The resulting phagosomes contain an arsenal of antimicrobial agents that then kill and degrade the internalized microbes. These agents include antimicrobial proteins and peptides, including defensins, catalysides, low pH, acid-activated hydrolytic enzymes, which includes lysozymes and proteases, and specialized molecules that mediate oxidative attack. Oxidative attack on the phagocytized microbes, which occurs in neutrophils, macrophages, and dendritic cells, employ highly toxic reactive oxygen species ROS and reactive nitrogen species RNS, which damage intracellular components. The reactive oxygen species are generated by the phagocytes' unique NADPH oxidase enzyme complex, which are called as phagosome oxidase, which activated when macros bind to the phagocytic receptors. The oxygen consumed by the phagocytes to support ROS production by NADPH oxidase is provided by metabolic process known as the respiratory burst, during which oxygen uptake by the cell increases sevenfold. NADPH oxidase converts oxygen to superoxide ions. Other ROS generated by the action of additional enzyme are hydrogen peroxide, that is H2O2, and hypochlorous acid, that is HClO, the active components of the household bleach. The generation of reactive nitrogen species requires the transcriptional activation of the gene for the enzyme inducible nitric oxide synthase, which is often called as INOS or NOS2, that distinguish it from related nitric oxide synthate in other tissues. The expression of inducible nitric oxide synthase is activated by microbial components binding to various pattern recognition receptors. The inducible nitric oxide synthase oxidizes L-arginine to yield L-citrulline and nitric oxide, a potent antimicrobial agent. In combination with a superoxide ion generated by NADPH oxidase, nitric oxide produces an additional reactive nitrogen species, peroxynitrite, and toxic S-nitrosothiose. Collectively, the ROS and RNS are highly toxic to phagocytized microbes due to the alternations of microbial molecules through oxidation, hydroxylation, chlorination, nitration, and S-nitrosylation, along with formation of sulfonic acids and destruction of iron-sulfur clusters in proteins. One example of how these oxidative species may be toxic to pathogens in oxidation by ROS of the cysteine sulfahydrins that are present in the active sites of many enzymes. Inactivating the enzymes ROS and RNS also can be released from the activated neutrophils and macrophages and kill extracellular pathogens. Evidence from genetic defects in humans and mice highlights the critical roles of these reactive chemical species in microbial elimination by phagocytic cells. The importance to antimicrobial defense of a phagosomal NADPH oxidase and its products, reactive oxygen species and reactive nitrogen species, is illustrated by chronic granulomatous disease, that is CGD. Patients affected with this disease have dramatically increased susceptibility to some fungal and bacterial infections caused by defects in subunits of NADPH oxidase that destroys its ability to generate oxidizing species. In addition, studies with mice in which the genes encoding inducible nitric oxide synthase were knocked out have shown that nitric oxide and substances derived from it account for much of the antimicrobial activity of macrophages against bacteria, fungi, and parasites. These mice lost much of their usual ability to control infections by such intracellular pathogens as Mycobacterium tuberculosis and Leishmania major, the intracellular protozoans parasites that causes leishmaniasis. As the body's main scavenger cells, macrophages also utilize their phagocytic receptors to take up and clear cellular debris, cells that have died from the damage or toxic stimuli, which are called as necrotic cell death or from programmed cell death or apoptosis, and aging red blood cells. Collectively, the components of dead or dying cells and damaged tissues that are recognized by pattern recognition receptors leading to their clearance are sometimes referred as damage associated molecular patterns or DAMs. As the presence of these components may also be an indicator of conditions harmful to the body or may contribute to harmful consequences such as autoimmune diseases, the D in DAM also can be referred as danger signal. Phagocytosis is the major mode of the clearance of cells that have undergone apoptosis as part of developmental remodeling of tissues, normal cell turnover, or killing of pathogen infected or tumor cells by innate or adaptive immune responses. Apoptotic cells attract phagocytes by releasing the lipid mediator lysophosphatidic acid, which functions as a chemoattractant. These dying cells facilitate their own phagocytosis by expressing on their surface a number of molecules not expressed on the healthy cells, including phospholipids such as phosphatidylserine and lysophosphatidylcholine, proteins, and altered carbohydrates. These DAMs are recognized directly by phagocytic receptors such as the phosphatidylserine receptors and scavenger receptors, that is SRA1. Other DAMs are recognized by soluble pattern recognition molecules that functions as opsonins, including the collectins, MBL, SPA, and SPD, various complement components, and the pentraxins, C-reactive protein, and serum amyloid protein. These opsonins are then recognized by opsonin receptors, activating phagocytosis and degradation of the apoptotic cells. An important additional activity of macrophages in the spleen and those in the liver, which are known here Kupffer cells, is to recognize phagocytosis and degrade aging and damaged red blood cells. As these cells age, novel molecules that are recognized by phagocytes accumulate in the plasma membrane. Phosphatidylserine flips from the inner to the outer leaflet of the lipid bilayer and recognized by phosphatidylserine receptors on phagocytes. Modifications of erythrocyte membrane proteins have also been detected that may promote phagocytosis. Clearly, it is important for normal cells not to be phagocytosed, and accumulating evidence indicates that whether or not a cell is phagocytosed is controlled by sets of eat me signals, the altered membrane components, that is DAMs, and don't eat me signals expressed by normal cells. Young, healthy erythrocytes avoid being phagocytosed by not expressing eat me signals such as phosphatidylserine, and also by expressing don't eat me signals, the protein CD47. CD47 expressed on many cell types throughout the body is recognized by SIRP alpha receptor on macrophages, which transmit signal that inhibit phagocytosis. Research studies have shown that tumors use elevated CD47 expression to evade tumor surveillance and phagocytic elimination by the immune system. Increased expression of CD47 on all or most human cancer is correlated with a tumor progression, probably because of CD47 activities. SIRP1 alpha mediated inhibition of the phagocytosis of tumor cells by macrophages. This understanding of the role of CD47 in preventing phagocytosis is being used to develop novel therapies for certain cancers, such as using antibodies to block CD47 on tumor cells, which should then allow them to be phagocytosed and eliminated. So friends, in this way, phagocytic barriers clears once the pathogen enters into the body. Next lecture we will learn the other components of innate immunity. Thank you.